So the ADA protein structure can be divided into 4 different sublevels. So primary is the simplest form, secondary, tertiary, and quaternary. Quaternary is referring to when there are 2 or more protein molecules being linked together to form a larger functional protein complex. So the ADA enzyme can either exist as a monomer, which is shown here, meaning there's only a single polypeptide chain making up the enzyme, or as a dimer, which is 2 of the polypeptide chain being combined. So we will only focus on the monomeric form because human ADA more commonly exists in this form. So first, let's talk about the primary structure. This is referring to a single amino acid chain or polypeptide chain. Poly means multiple, and peptide refers to an amino acid residue, which is shown here. Each amino acid is linked together through a type of bond, which is called a peptide bond, which is basically formed between an amino group of one amino acid residue and a carboxyl group of another residue, forming what is called the backbone of the polypeptide chain. So the 2 ends of the polypeptide chain is called the N-terminal, which is the end that has an amino group, and the C-terminal, which ends with a carboxyl group. And the amino acid sequence is almost always written from the N-terminal, which is from the left, to C-terminal, which is to the right. And each amino acid has the same backbone structure, which is highlighted here in blue. However, they differ by the side structure, also known as the R group. The R group of different amino acid residues can interact with each other to form the 3D shape of a protein, which will be discussed later in this video.